Hi, my name is Mortaza Razavi. I am the Director of Lab Operations at SISCAPA Assay Technologies, and today I'll be presenting the automated SISCAPA workflow that we've developed in collaboration with Agilent Technologies. The workflow utilizes a Bravo liquid handling robot to automate digestion, SISCAPA enrichment, wash, and elution processes. If you have any questions about the workflow or require more information about SISCAPA, please visit us at www.siscapa.com. Before I get into the automated SISCAPA workflow, I think it might be a good idea to give you a brief overview of the SISCAPA technology itself. SISCAPA stands for Stable Isotope Standards and Captured by Antipeptide Antibodies. And the process of developing a de novo SISCAPA assay begins by selecting a protein biomarker that you're interested to measure in a biospecimen, which can be as simple as a cell lysate or as complex as human blood. We employ a proprietary algorithm to select one or multiple proteotypic peptides that are unique to that protein biomarker, and therefore measuring the specific peptide serves as a surrogate for measuring the protein itself. The selected peptide is then coupled to a carrier molecule for immunization. After years of experimentation, we have settled on rabbits to make our antibodies because in our experience, they produce high-affinity antibodies at a higher frequency compared to other host organisms. Another advantage of using rabbits is that they only produce one subclass of IgG, and this is very important when you want to generate homogeneous, consistent antibody reagents. After the immunization process, the hybridoma clones that are generated go through an exclusive screening process to select antibodies with sub-nanomolar affinities. Once you have your antibody reagent, you can make accurate measurements of your target peptide. This process begins by digesting your protein sample, which is usually done in a 96-well plate format. To this plate, you then add your highly selective antipeptide antibodies, which are attached to magnetic beads. At the same time, you will add an internal standard in the form of stable isotope version of the target peptide, which is shown in red in this presentation. I should also mention that this process is amenable to multiplexing. In other words, you can make simultaneous measurements of multiple analytes without losing any sensitivity or specificity. During the enrichment process, the antibody captures both the endogenous peptide and the stable isotope peptide with the same affinity. The ratio of endogenous peptide to cispeptide is used to calculate the concentration of the target protein you're interested in. Now that we've covered the technology, let's go over the details of the assay prep. We have divided the procedure into three segments: digestion, enrichment, wash, and elution. The first step of the quantitation process is to create the peptide fragments that will be enriched and measured. To do this, we follow an addition-only philosophy in order to generate target peptides in a reproducible manner with utmost sensitivity. The process does not involve any kind of separative techniques such as solid phase extraction or centrifugation or what have you. The second step is enrichment. The high-affinity antibody allows us to enrich the target peptide from a much larger sample volume than typically used in SRM assays. Also, because of the enrichment process, the matrix that is injected into the mass spectrometer is significantly simplified, which dramatically reduces ion suppression effects. Putting these two facts together, we reach a level of sensitivity that is not matched by any other mass spec assay out there. Finally, we have the wash and elution processes. We wanted to generate an eluate that is 99.99% pure so that we can analyze the samples with minimal chromatography, or in some cases, no chromatography at all. And we reached that goal using this automated workflow. So let's go over the workflow in more details. This is the layout of the Bravo deck for the first step of the workflow, the digestion step. In the first position, we have the tips out container, which is used to discard reagent tips. Reagent tips are in position 2. These are tips that are used to handle digestion and SISCAPA reagents. In position 3, we have our digest plate. This is the plate that contains the denaturation mix and where your protein sample will be digested. The sample tips are in position 4. As the name suggests, these are tips that transfer the samples, which are in the red 96-well plate at position 5, to the digest plate. At position 6, we have an 8-row reservoir plate that contains the digestion reagents. These are the alkylation solution and the enzyme that you use for digestion, which is trypsin in most cases. The diluent tips are in position 7. These tips transfer the diluent from the reservoir plate at position 8 to the master plate at position 3. Okay, now it's time to see the whole process in action. The process begins by grabbing the sample tips and transferring samples from the red sample plate to the digest plate. The digest plate originally contains the denaturation reagent, which will be incubated with the samples by shaking the plate at room temperature for 30 minutes. Next, one row of reagent tips is selected to transfer the alkylating solution to the digest plate. This is done in a row-wise fashion. The height at which the tips dispense the reagents is adjusted such that the tips do not touch the samples. At the end of this process, the tips are discarded in the tips out container. The plate is shaken to mix the reagents, and we have a 10-minute incubation at room temperature without shaking. Next, the diluent tips are grabbed to transfer the diluent to the digest plate. Finally, another row of the reagent tips are selected to transfer trypsin to the master plate. After addition of trypsin, the plate shakes for 60 seconds at the end of the process to mix the entire solution. At this point, we transfer the plate to an incubator at 37 degrees Celsius for digestion to take place. Digestion time for most proteins using this approach is 1 to 3 hours. However, for de novo assays, this variable needs to be optimized. While the sample is digesting, we reshuffle the Bravo DEX to prepare it for step 2 of the workflow, which is the SISCAPA enrichment step. In this picture, we see the layout for step 2. There are no changes to the first row. We still have the tips out container in position 1, reagent tips in position 2, and the digest plate is still on the shaker at position 3. At position 4, we have the cis tips, which are used to transfer the stable isotope peptides from the red 96-well plate in position 5 to the digest plate. Finally, at position 6, we have a 96-well plate holding the SISCAPA reagents. These are a solution that stops trypsin activity and the antibody beat complexes. Before enrichment, trypsin activity needs to be stopped. The robot takes one row of reagent tips to transfer the trypsin inhibitor solution to the digest plate. The tips are discarded and the plate is shaken for 60 seconds. The plate has to incubate for 5 minutes at room temperature before proceeding. Next, cis tips are used to transfer the stable isotope peptide mixture to the digest plate. Finally, another row of reagent tips are selected to transfer the antibody beat complex to the plate. The robot first mixes the antibody beat complex by pipetting the solution up and down for 20 times. The tips are discarded and the plate starts to shake. The enrichment process takes place by shaking the plate at room temperature for 60 minutes. While enrichment is taking place, you have time to reshuffle the deck once more to get ready for the last step. This time, we have the recovery tips in position 1. These are the only tips that come in contact with the digest during this process. We have a 96-well plate in position 2 that will hold the final eluate at the end of the process. This is the plate that will be analyzed by the mass spectrometer. Digest plate is still on the shaker at position 3. At position 4, we have a clean plate. The antibody beat complex is transferred to this plate after the second wash. This is important for reducing the non-specific background as much as possible. We have a 384-well plate at position 5, which holds 3 wash buffers and 1 elution buffer for each well of the digest plate. At position 6, we have the recovery plate. We save the digested sample in this plate in case we want to further analyze the samples, perhaps for different analytes. At position 7, we have the wash tips. These tips are the only tips that will handle fresh wash buffers during the process. At position 8, we have the elution tips, and finally, at position 9, We have a magnet plate that is used to pellet the antibody bead complex during this procedure. The plate is specifically designed to pellet the beads in the shortest amount of time possible. Before washing the antibody bead complex, the digest needs to be removed from the plate. The plate is therefore transferred to the magnet plate where the beads are pelleted and the recovery tips are used to save the digest in the recovery plate. Next, the wash tips are used to add wash buffer to the master plate while it is still on the magnet. The plate is then transferred to the shaker so that the beads can be resuspended. This process takes 60 seconds. After the shaking, the plate is once more transferred to the magnet so that the used wash buffer can be removed using the recovery tips, and fresh wash buffer can be added once more using the wash tips. After the second wash, before removing the second used wash buffer, the content of the master plate are transferred to the clean plate. The clean plate, which is now our main plate, is then transferred to the magnet, so that removal of second wash and addition of the third and final wash can take place. The third wash is now added. The robot transfers the old digest plate to position 6, and that empties the shaker to be used by the clean plate. So now the clean plate is getting transferred to the shaker. The antibody bead complex is going to be resuspended in the third wash, and the process of removing the third wash is going to be the same as washes 1 and 2. Once the third wash is removed, The elution tips are used to add elution buffer to the plate. We then shake the plate for 3 minutes for elution to take place, transfer the plate back to the magnet so that we can pellet the antibody bead complex, and finally use the elution tips to transfer the eluate to the final 96-well plate, that is the red plate at position 2. And so that is the end of the process. The red plate is the product plate that is going to be analyzed by the mass spectrometer. Before I finish the presentation, I should mention that this is the beta version of the SISCAPA workflow. Future iterations will include other Agilent robotic instruments to integrate all 3 steps into one walk-away type protocol. On that note, thank you for watching, and please do not hesitate to contact us should you have any questions.